Hello, everybody, teachers, classmates, and to all listeners out there, good morning. So, for today's topic, let's talk about nucleic acid. This is your reporter, Maureen Balogan, and let's talk about the types and building blocks of nucleic acid. A most remarkable property of living cell is their ability to produce exact replicas of themselves. Furthermore, cells contain all the instructions needed for making the complete organism of which they are a part. The molecules within a cell that are responsible for those amazing capabilities are no other than the topic itself, the nucleic acid. So before proceeding to the lesson, let us first find the definition of the topic at hand. So what is a nucleic acid? It is a class of biomolecules that are found in all living organisms, a term used for the class of biological polymers consisting of deoxyribonucleic acid and ribonucleic acid. It is responsible for the storage of genetic information. They are also responsible for protein synthesis. The Swiss physiologist Friedrich Miescher discovered nucleic acid in 1869 while studying the nuclei of white blood cells. The fact that they were initially found in cell nuclei and are acidic accounts for the name nucleic acid. Although we now know that nucleic acid are found throughout the cell, not just in the nucleus, the name is still used for such materials. There are two types of nucleic acid which are found within cell of higher organism. They are the deoxyribonucleic acid, or also known as DNA, and ribonucleic acid, which is also known as RNA. A deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instruction for the development, functioning, growth, and reproduction of all known organisms and many viruses. Nearly all the DNA is found within the cell nucleus. Its primary function is the storage and transfer of genetic information. This information is used indirectly to control many functions of a living cell. In addition, DNA is passed from existing cells to new cells during cell division. Ribonucleic acid, on the other hand, is a molecule similar to DNA. Unlike DNA, RNA is a single-stranded. It is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expressing of genes. RNA occurs in all parts of a cell. It functions primarily in synthesis of proteins, the molecules that carry out essential cellular functions. In general, all nucleic acid molecules are polymers. A nucleic acid is a polymer in which the monomer units are nucleotides. Thus, the starting point for discussion of nucleic acid is an understanding of the structures and chemical properties of nucleotides. Let us now move on to the building blocks of nucleic acid, which is the nucleotides. Nucleotides is a 3-subunit molecule in which a pentose sugar is bonded to both phosphate group and a nitrogen-containing heterocyclic base. With a 3-subunit structure, nucleotides are more complex monomers than the monosaccharide of polysaccharides and the amino acid of proteins. The picture presented represents the block structural diagram of a nucleotide. The sugar unit of a nucleotide under the 5-carbon sugar is either the pentose ribose or the pentose 2'-deoxyribose. Under the phosphate functional group lies the component of nucleotide which is derived from the phosphoric acid. And lastly, the nitrogen-containing heterocyclic base, which are composed of adenine, guanine, cytosine, and thymine. Now, let us pay attention to the pentose sugars. Structurally speaking, the only difference between these two sugars occurs at carbon 2'. The carboxyl group present on this carbon in ribose becomes a hydrogen atom in 2' deoxyribose. So take note that the prefix deoxy means without oxygen. RNA and DNA differ in identity of the sugar unit in their nucleotides. In RNA, the sugar unit is ribose, hence the R in RNA. In DNA, on the other hand, the sugar unit is 2' deoxyribose, hence the D in the DNA. Moving on to the nitrogen-containing heterocyclic bases, there are 5 nitrogen-containing heterocyclic bases which are nucleotide components. 3 of them are derivatives of pyrimidine, a monocyclic base with a 6-membered ring, and 2 are derivatives of purine, which is a bicyclic base with fused 5 and 6-membered rings. As you can see here in the picture, both of these are heterocyclic compounds which are bases because they contain amine functional groups, which is either secondary or tertiary, and amine functional groups exhibit basic behaviors. The 3 pyrimidine derivatives found in nucleotides are thymine, cytosine, and uracil. Thymine is the 5-methyl 4-deoxyderivative, cytosine is the 4-amino oxiderivative, and the uracil as 4-deoxyderivative of pyrimidine. The 2 purine derivatives found in nucleotides are adenine and guanine. As represented in the picture, adenine is the 6-amino derivative of purine, and guanine, on the other hand, is the 2-amino 6-oxopurine derivative. Adenine, guanine, and cytosine are found in both DNA and RNA. Uracil is found only in RNA, and thymine usually occurs only in DNA. That concludes my report. Thank you for listening. To further understand the lesson, continue listening for the next part.